Have you ever felt a longing so deep, a pull so insistent? It felt like everything within you was reaching for something just beyond your grasp. Poets have written about it for centuries, capturing the ache of unrequited love in stories like Romeo and Juliet, Tristan and Isolde. But what if I told you that same fundamental drive, the same insistent pull, is happening within you, right now, in every single movement you make? It's not a story of star-crossed lovers in Verona, but a microscopic drama unfolding in your muscles. A relentless coupling and separation, a constant dance between two tiny protein strands, actin and myosin. Forget tragic endings. This is a story of life itself. This is the engine of motion, the force behind every step, every breath, every word you speak. Think about it. Every gesture, from the subtle flicker of an eyelid to the powerful stride of an athlete, is powered by this microscopic interaction. Your muscles aren't just supporting your weight against gravity, they are actively defying it, moment by moment. They are tireless workers, constantly converting the potential energy within you, the energy from the food you eat, into the kinetic energy of movement. And that transformation hinges on the intricate relationship between actin and myosin. Imagine a world built on attraction and release. That's the essence of muscle function. Muscles contract, they shorten, they pull. And then they relax, they lengthen, they release. A continuous cycle, a rhythmic ebb and flow, all driven by the elegant simplicity of these protein interactions. It's a beautiful, fundamental process. Now let's zoom in. You have three main types of muscle tissue: smooth, cardiac, and skeletal. Smooth muscle lines the walls of your internal organs, your stomach, your airways, your blood vessels. It works tirelessly, without you even knowing it, pushing fluids and materials along their paths. It's the unsung hero of internal regulation, a constant, quiet force. Then there's cardiac muscle, the specialized tissue of your heart. It's striated, meaning it has a banded appearance and it beats relentlessly, pumping life-sustaining blood throughout your body. Again, involuntary, automatic, essential. You don't have to tell your heart to beat, it just does. But when someone says muscle, what image comes to mind? Probably the sculpted physique of an athlete, the defined contours that represent strength and dedication. Those are your skeletal muscles, the ones you consciously control. There are 640 of them, responsible for everything from walking and running to writing and painting. Like cardiac muscle, they're striated, showcasing the organized arrangement of their internal structures. Each skeletal muscle isn't just a bundle of fibers, it's a complete organ in itself. A complex structure composed of muscle tissue, connective tissues that provide support and protection, a network of blood vessels to deliver oxygen and nutrients, and nerves that transmit the signals to move. Think of it as a highly engineered machine, perfectly optimized for its task. And those muscles are hungry. They demand a constant supply of energy. That's why each muscle is supplied with its own dedicated nerve to initiate contraction, and its own artery and vein to ensure a steady flow of fuel. They're not just powerful, they're metabolically demanding. To truly understand how muscles work, we need to delve into their anatomy to appreciate the layers within layers. Picture a sturdy rope. That's similar to how a muscle is constructed. Thousands of tiny, parallel threads called myofibrils are bundled together to form muscle fibers, the actual muscle cells. These fibers are remarkably complex, containing mitochondria, multiple nuclei, and a cellular membrane called the sarcolemma. These muscle fibers aren't isolated. They combine into larger bundles called fascicles, and these fascicles then intertwine to form the complete muscle organ, like your biceps brachii or your gluteus maximus. This nested structure gives muscles their strength and resilience. But even with this robust architecture, muscles are subjected to tremendous stress. Lifting heavy objects, enduring intense exercise, these actions put a strain on the muscle tissue. That's why they're reinforced with connective tissues, acting as protective sheaths to prevent damage. So we've covered the structure. Now let's get to the heart of the matter. How does a muscle actually contract? How does this microscopic dance between actin and myosin translate into movement? There are two fundamental rules governing this process, rules that apply to proteins throughout your body. First, proteins are dynamic. They change shape when other molecules bind to them. Think of it like a lock and key. The key alters the lock's configuration. Second, and crucially, a change in shape can either promote or prevent further binding. It's a delicate balance of attraction and repulsion. Keep those rules in mind as we journey deeper into the sarcomere, the functional unit of a muscle fiber. A sarcomere is divided into regions containing two key players: actin filaments, which are thin and twisty, and myosin filaments, which are thicker and more club-shaped. These filaments aren't randomly arranged. They're organized in a precise pattern, delineated by structures called Z-lines. When a muscle contracts, these Z-lines draw closer together, shortening the sarcomere. Now the romance begins. Imagine actin and myosin wanting to connect, to interlock. That's the driving force behind muscle contraction. This interaction is described by the sliding filament model. But like any compelling love story, there are obstacles. Actin is initially blocked by proteins called tropomyosin and troponin, acting as gatekeepers, preventing myosin from attaching. But these gatekeepers aren't impenetrable. They can be bribed with ATP, adenosine triphosphate, the energy currency of the cell, and calcium ions. Think of ATP as the fuel that powers this entire process. Your muscles are constantly consuming ATP to maintain their activity. That's why they're packed with mitochondria, the ATP-generating factories of the cell. Muscle cells also have a specialized version of the endoplasmic reticulum, the cell's internal transport system, called the sarcoplasmic reticulum. Its walls are lined with calcium pumps, tirelessly working to store calcium ions. Simultaneously, calcium channels are embedded in the sarcoplasmic reticulum, linking it to voltage-sensitive proteins on the muscle cell membrane. Let's trace the journey of a signal. When you decide to move your arm, your brain sends an electrical impulse, an action potential, along a motor neuron. This neuron synapses with a muscle cell in your arm, releasing acetylcholine into the synapse. This neurotransmitter binds to receptors on the muscle cell, opening sodium channels and initiating a rush of sodium ions into the cell, a graded potential. If this signal is strong enough, it triggers the opening of voltage-gated sodium channels, amplifying the signal and propagating it along the muscle cell membrane, the sarcolemma. And this is where things get really interesting. The sarcolemma is riddled with T-tubules, deep invaginations that carry the action potential into the heart of the muscle fiber. As the action potential travels down these tubules, it triggers the voltage-sensitive proteins linked to the calcium channels in the sarcoplasmic reticulum. These channels open, unleashing a flood of calcium ions into the cell. And that's the signal myosin has been waiting for. The calcium ions bind to troponin, causing it to shift position and pull tropomyosin away from the myosin-binding sites on the actin filament. It's like removing a barrier, opening the door for connection. The myosin heads, which have already been primed by breaking down ATP into ADP phosphate, are now free to bind to actin. When myosin and actin finally connect, it's a moment of explosive energy release. The myosin head pivots, pulling the actin filament along with it, shortening the sarcomere and contracting the muscle. This is the sliding, part of the sliding filament model. The myosin head, having expended its energy, releases ADP phosphate, creating a space for a new ATP molecule to bind. This binding triggers another shape change, causing the myosin head to detach from actin. It's a heartbreaking moment, a microscopic recreation of the final scene in Titanic. But the cycle isn't over. This detached myosin head is immediately re-energized by breaking down another ATP molecule, returning it to its primed position, ready for another round. Meanwhile, the calcium pumps are working tirelessly to remove calcium from the cytoplasm, restoring the tropomyosin and troponin to their blocking positions. The entire process repeats itself, over and over again, hundreds or even thousands of times per second. It's a relentless, coordinated effort, orchestrated by the interplay of proteins, ions, and energy. The next time you take a step, lift an object, or even blink an eye, remember this microscopic ballet unfolding within your muscles. It's a reminder that even the most complex movements are built on the foundation of simple, elegant interactions. And it's a testament to the incredible power and efficiency of the human body. This isn't just about muscle contraction. It's about life, movement, and the fundamental drive to connect, to reach, to act. So appreciate the power within you, the tireless work of your muscles, and the beautiful, microscopic romance of actin and myosin. It's a story worth remembering. Remember this: your smooth, cardiac, and skeletal muscles create all movement through the process of contraction and relaxation, driven by the sliding filament model. Recall that your skeletal muscles are constructed like a rope of protein fibers, with actin and myosin filaments at the core. And understand that the binding and unbinding of these filaments, fueled by calcium and ATP, is the key to life, movement, and the very act of being.